Everybody, next topic is supramolecular catalysis. In catalysis, the catalyst are directly does not involved in the chemical reaction, but it alter the path by stabilizing the transition state. So in supramolecular chemistry, supramolecule act as a catalyst for many of the chemical reaction, and it bind the reactant molecule in their active site, and after the chemical reaction it will liberate the product molecule, and the catalyst is regenerated after the chemical reaction. Now we are moving into the some of the mechanism of the catalysis of the supramolecule. For every catalytic process, the reactant molecule must have a particular orientation, and the concentration of the reactant molecule in the active site of the catalyst is very important. So in supramolecular chemistry, the reactive molecule or substrate molecule is binded to the host supramolecule as a gust molecule, and the gust group is positioned very close to the reactive group. So due to this, there is a better probability for the chemical reaction, and effective substrate concentration is also increases. So the bimolecular reaction naturally occurs inside the molecule. This is mainly due to in the binding site, the concentration, the binding site will bind the reactive molecule, and there is an interaction is existing between the reactant molecule, and so the normally the rate of a reaction is accelerated, and this is mainly due to the concentration is increases inside the macromolecule, inside the supramolecule, and there is a intermolecular reaction is faster in the supramolecular chemistry than the corresponding intermolecular reaction, and the supramolecule stabilizes the transition state in the catalytic process. For example, a Diels-Alder reaction between the two pyridine functionalized substrate normally yield a mixture of the exo and endo product. But if you are using the supramolecular catalyst, we will get only one product. Either it may be exo or endo product. Now, as we discussed earlier, the supramolecular catalytic reaction involving the binding of well-defined substrate, that means the reactant, to the receptor, that is the catalyst. And there are three steps are involved in the supramolecular catalysis. First is the selective binding of the reactant in the supramolecule, and the next is the transformation of the bound species. Third one is the release of the product and regeneration of the catalyst. So the first two steps take part in the molecular recognition, and the substrate are inserted into either an active site of enzyme or a host cavity or a layer or a micelle or a vesicle or a micropore of the supramolecule. So the functional group in the active site or the cavity, such as hydrogen bonding donor or an acceptor group, can additionally exert the true catalytic action. Now, the example for the catalysis by the reactive macrocyclic cation receptor molecule, in which the amino group primary ammonium ion is binded in the 18,6 macrocyclic polyether, and after the reaction the product the macromolecule liberate the product and it is regenerated. The next is the catalysis by reactive anion receptor. As you know that anions are relatively larger and therefore require larger receptors than that of cation. And one of the smaller anion is F minus, but it is the size of the F minus is comparable to the ionic radius of the K plus, that is 1.33 angstrom for F minus, and for K plus it is 1.38 angstrom. So the inorganic anions occur in a range of the shape and geometry. For example, the halides are spherical in shape, but SCN N3 minus are linear in shape, NO3 minus are planar in shape. So depending upon the shape and geometry of the anion, there are various receptors for binding these anions. And anions have high free energy of the solvation, and anion are bind via weak forces such as hydrogen bonding and van der Waals interaction. This is the one of the example for the catalyst which are using anion receptors. Here the macromolecule using the ether and using the nitrogen and oxygen binder is used for binding the phosphate group in the macromolecule, and the weak hydrogen force and is operating here for binding the anion. After the product formation, it will regenerated. Catalyst is regenerated.